You can do it with amino acids. You've got to be able to do it with DNA, and that means you need to know them. Whether they're RNA or DNA, you won't know, but you'll have to draw them. So you got to learn all of them. Oh, no problem. It's actually not that bad. Okay, so again, this is not as long a list as chapter 7, right? We're just sort of flying through this, and if we were even filling in here, this is more emphasized than even chapter 9. So, all right, put all that away, and we'll see what we should learn today. See what else is oh, someone might be asking the question, and I'll fix it, but not what I wanted, but that's okay. The upcoming extra credit assignment, when did it move to?
Saturday.
Good. And I actually thought I removed it, but there's still a fake link that leads you out to feedback fruits. I removed the feedback fruit. It's all built, but I removed that. I'm just gonna grade them. So just upload them right into Canvas. It'll send you back in and it says start assignment. It should let you submit it as an upload document. So that's really what you're doing. I'll try to get that fixed so that that's not an issue and you're running into it, but if you're confused, you're getting it to the right place if it's uploading right in Canvas. Should be good. Okay. Those are some pretty pictures, right? There'll be a pattern in all things, that ye may not be deceived as Satan's abroad. Where do we get the power? He that prayeth, whose spirit is contrite, the same is accepted of me, if he obey my ordinances. Is obedience important to the Lord?
Yes.
It's the first law of heaven. It gets us blessings. Are any of us going to be perfect? No, we're not. But are there things we need to do to get the blessings that we're wanting? So one day, what do you have to be able to do? If you're going to be like Heavenly Father, what do you actually one day need to have happen? You'll need to be perfect. It's not going to happen in this lifetime. I mean, even the Savior himself didn't say that he was perfect when he was here on earth. Okay? Well, if that's the high bar that we're trying to get there, sometimes we freak out about it. It's like, I will never get there. Please recognize that if you'll keep doing the right things, stay on the straight and narrow, the Lord can help you change your heart. It will come in time. Okay? Don't give up on yourself. Don't be depressed. Don't be discouraged. You can do this. But also don't continually make excuses. Sin is sin. And what you're really losing out on is the power that the Spirit can bring into your life. And it's that very power that helps you overcome those very weaknesses you're trying to get over. So invite the Spirit more. Partake of the ordinances. Move forward. Be in the temple. Read the scripture. You want the Spirit because it's power. Okay. It's a nice list of fun things to look at. Let's look at some biological functions of it. ATP. Nice. For a long time in teaching high school biology, I had no idea what ATP stood for. Isn't it funny now? In my reflection, I'm like, oh, of course, adenosine triphosphate, right? You know that word. What does that mean? What does that molecule mean? How does it look? How does it look to the body? Presented to the very same things you just have been studying. We keep looking at these classes of biological molecules. This is another class of biological molecules, and they are special. These are the ones that code for life, right? Are they used in other places? Of course. NAD, as in NADH. What's that used for? Anybody know? Yeah.
Synthesis and
to make the ATP, it's a high energy molecule. It'll accept a hydride and then we'll transfer it. We use NADPH too. It's just a substitute with a phosphate on it. FAD, by the way, also has it. It's also a nucleotide. CAMP, well, it's obviously made from ATP. It's a cyclic one, but that becomes a secondary messenger that we use heavily, right? Your mRNA, storing your DNA, the ribosome itself is built out of what? RNA, that's interesting. tRNA, RNA for protein synthesis. Okay, so we have a lot of other uses besides just the code. And so it is of value to us. These are basic definitions, make easy exam questions that are low level, easy ones you want to grab. A nucleotide consists of 3 pieces. The nitrogenous base itself, a pentose, the os ending indicating that it is sugar. In fact, generally speaking, what's this sugar?
It's right.
It's a ribose.
Yes.
Is it the deoxyribose or not?
Yes.
Tell your neighbor why you know it is or isn't.
That's normal ribose. Yeah, well, it's got, if you look at the second carbon specifically, it's got that hydroxyl bond. Deoxy would be missing that one.
Is it accidental that it's in beta position for the nitrogenous base link? It's always that way. How can it do that? If it can go either alpha or beta, how does it do it? What must be controlling that?
Enzymes.
Enzymes. It's a beautiful thing. Enzymes are very powerful. And then the phosphate attached. So you've got these 3 pieces that make up a nucleotide, but if I call it a nucleoside, well then it's missing something. What was it missing? A phosphate off the side gets pulled off for the nucleoside. And a nucleobase is just, well, that one's obvious to us. But these two can be confusing, okay? So figure out some mnemonic, some little trick. How are you gonna remember which is the difference between the tide versus the side? But the one's just missing the phosphate group, okay? Pyrimidine versus purine, you should know those in your functional vocabulary. These structures are good to study this way, but actually I'm gonna show you it's better to study it one a little bit lower, and we'll practice that in just a second, okay? pH, what do you think? Are they gonna be negative charge? They're very negative.
Yeah.
Yeah, I mean, for all intensive purposes, we just wish they were more positive with the outcome of our life, right? I mean, but no, they're really negative about it. Yeah, that was. Typically attached to the 5', we do the triphosphate versions. Coincidentally, you'll learn later as you take the 2nd semester of biochemistry, all of these are interchangeable. I can change the nucleobase in all of those, and there's an enzyme that for 0 charge can convert any one of those into the other. So if I'm short on ATP, I can pull it from any one of those others, and they're all the equivalent energy source. Okay, they could be attached to other positions. Some of you are starting registration. If you are looking at next semester, 482 is offered winter semester and fall semester only. That is the 2nd semester of biochem, dealing with metabolism. I just looked at several references on it again because I had one of our biochemistry majors who asked the question because he was getting advice from academic advisement as a biochemistry major in his last 2 semesters that he should probably make it look better if he took his Nat Phys next semester rather than biochem. That's a loaded dice. But the obvious thing is, since it's not offered in the spring, he's not graduating, not going to medical school if he doesn't take it next semester, right? If you were just not having to worry about that, and you still had 2 semesters left, I don't know that I would pick Anat Phys over biochem. What is the most challenging, what is the most flunked course in medical school? It is second semester biochem. That's where you start on semester 1. It will be the one that crushes many.
Yay.
But it doesn't have to be. It's really an undergraduate course, you guys. And it's the most challenging course in medical school. Imagine flunking it and having to spend another cost of medical school, $30,000 to $50,000 to go again, one more semester. That's like your whole undergraduate education just like blown out the window because you just like flunked a course. That's a cool course to take. I'd take that course. And if I were the one evaluating it on the admission side, I'd be looking at it going, this guy's passed our most challenging course, he'll probably pass our program. By the way, which is the second most challenging course? Anat Phys. They're the next one down.
Jesus Christ.
So somebody's got the wrong position, but they know what it's talking about. They're just missing the point. All right. But we don't all go medical school. We don't all. All right. Different positions. Awesomeness. Let's see. We've got the deoxy form. We've got that figured out. Beautiful. UV light. Tell somebody else you remember where UV light is absorbed if you are a protein. What wavelength are we interested in and why?
2, 4.
Yeah, it's because of the.
I remember like 2.
Yeah, it's
in my life. I remember how that shit was crap.
Yeah, but
I remember talking about it. It's interesting.
280. I know 80. 280. 380 doesn't make sense. It's 280. Yeah.
Is it?
I thought about 380 would be like.
I know, I'm like
244. Ultraviolet. I don't.
Did you name aromatics?
The other direction.
What were the 3 aromatics? Tryptophan, phenylalanine, and tyrosine. Which were the 2 that absorbed at one wavelength and the other at another wavelength? The 280 you're interested in are at?
That's
tryptophan and
phenylalanine.
tyrosine. And then the phenylalanine has a very low absorbance at 260. But this is also where your DNA shows up and your RNA because it's a heterocyclic aromatic. Okay? All right. So 260, is that used? Absolutely. You're purifying protein, you're going to look at that wavelength. See how pure you got it. And could you have contaminants? Absolutely.
So, out of curiosity, why exactly do aromatics interact with light so readily? Maybe that's more of like a physical chemistry question. But
No, but it's one you should be able to answer. Let's just go back to 105. Okay? That long ago thing you forgot. Okay?
it's been a while.
In order for light to be absorbed, what do I need? The electrons to encounter
Photons.
the light, the energy from the light, and then it would be excited to an upper level, right? If it was at the right wavelength. So if I have a pi system,
Delocalized electrons.
do I have a big target?
A very large area that light can interact with those electrons. Yeah.
That's correct. And what about their energy?
Yeah.
Easier to change that wavelength frequency up there. So big target, easier to change. So yeah, and if I really start changing them at the bond level beyond that in a non-aromatic, like a single bond, what's that going to eventually do to the molecule?
Break it apart.
That'll break it apart.
Yeah.
So does that help?
Yeah.
I think
It's
it's
a good review.
Yeah, good.
I don't remember this.
Oh.
No, I appreciate you just asking the question, because it's really valuable for all of us. And I'm sure others benefited from it. So let's actually pull out markers. Let's take a moment, whether on your paper, which is where you're gonna draw it, your notepad, where you're gonna draw. We're gonna draw this. Which one should we draw? Let's draw one like this, okay?
Find my.
Draw it on your sheet of paper, put it down,
Which one did you say?
choose by me. I'm waiting for you. You done yet?
That's right.
These are the,
Sorry. Nice to put the rest of it together. Okay, did you draw them? Are you, in looking at those structures, in trying to draw it, do you recognize that they're actually, the majority of it is the same? Is it really the majority over here still? Yep, the majority of it's all the same. So yes, you have to memorize them, but the basic backbone's virtually identical, just minor changes. And it isn't weird that there's an enzyme that puts on and off this right here. Coincidentally, we actually start with this, we put it on, so.
So we start with the uracil, is what you're saying?
Yep. In the deoxy form. Isn't that exciting?
Huh.
Okay. Don't put away your markers. Let's consider what's similar about these versus what you just drew. Leaving it on the same page, like, oh, wait. Is this similar? The answer is, of course it is. Get at least one of them drawn next to it. Can you see the similarities in the pattern? This basic, now what is this basic piece? Is this the purine or pyrimidine?
It's the purine. Pyrimidine. Thank
The
you.
pyrimidine is the basic block. What's the bigger one?
The purine.
Okay, so all right, so you're looking at the purine right now as the 2-piece ring cycle, but the pyrimidine base is really still found inside of that purine. Okay. Since you're gonna have to learn them and draw them, is that helpful to just take a moment to be thinking about that this is not as bad as it, because you're gonna probably start by drawing flashcards, but what you do need to be able to draw the basic core of it so that your flashcards work for you.
Yeah, it does help a lot. Thank you.
Okay. There's a glycosidic bond, and it's formed with a linkage to what? The nitrogen. But which position on the nitrogen? okay? The bond's always with an anomeric carbon off the sugar in the beta configuration. The bond formed with in the pyrimidine, the N1 position. Okay? Where must N1 position be? And in the purine, N9. There's a problem. The textbook draws them most commonly like this. Now, some of you've taken OChem. Tell me what is wrong with this structure here. Why is this drawn the way it is problematic? It can be there, by the way. It's just not common there. Yeah, what do you think?
Because the charging space is usually like it's a molecule.
Yeah, it'd want to separate. We call that steric interference. These electron clouds are getting in the way. I've got lone pairs that don't like this. So syn versus anti is what we're discussing on that. So we'll want to look at those. It can rotate, but which one does it prefer?
Anti.
The anti. So when you learn them, all the textbook will have them drawn in which way?
Same.
And which way should you be drawing them?
Anti.
Start practicing this way. Easy. Right from the get-go. Start practicing this way. Then you'll know that they're right. Okay? Just turn it around and do it the way it's supposed to. Okay? All right. Yeah. Tautomerization, exciting, don't really care. Awesome. Absorption, we talked about that matters. There is no fluorescence for the nucleic acids. There is for tryptophan and amino acids. Homo, lumo, you've heard this. What was this acronym standing for when I looked at these MO diagrams?
Highest occupied molecular
Highest
orbital.
occupied molecular orbital, and then
Lowest
lowest
unoccupied.
occupied molecular orbital.
Unoccupied.
This had a lot to do with, when we were describing it, the energies they're found in and absorption of light. Okay?
Did you just say no homo?
You can't just draw them and call them a nucleotide, a nucleoside, and a nucleobase. You've got to also handle naming. We had to name sugars, we've got to name these guys. Are there different names that, well, purines, adenine, guanine, pyrimidine, cytosine, thymine, uracil. So largely the ine ending is going to go with the pyrimidines and the purines. Okay? I know uracil's weird. And then what does uracil do? Messes with you, yeah. But here I now put the adenosine sign. And so the side is going with the sign. Okay? And then it's not so helpful down here. But hopefully you're getting the idea. Adenylate, the 8 ending is the ones that are across the board on that. So 8888. It's your nucleotides. Yeah.
So when we talk about ATP being adenosine triphosphate, adenosine being the nucleoside, which means it doesn't have this phosphate group in the mix. That means because we identify the triphosphate.
Yeah, isn't it a funny thing? We're describing the adenosine triphosphate separate because we're describing the triphosphate we're naming.
On
it.
site, because it's also
Yeah.
included.
Yep.
Okay.
That's why. Is there single letters? Of course.
Yep.
2 letters? Of course. 3 letters? Of course. But generally speaking, you just need to know the D means what? Deoxy, and either A or AMP or GMP, you know, that's what you're looking at. So please know what these are you're looking at. These are the structures, and they're drawn not incorrectly, but in the form that you don't want to learn them. You want to, that's the sin position, turn them to the anti, and you'll be happier. Same here. Beautiful. You're like, oh, that's 8 structures. Is it really? Is it really? No, what's the only one different between these? Yeah, so if I learned one set here, and I knew all my deoxy form, then I would know the thymidine, and then I'd turn around here, and I need to know the uridine. Okay, just swap that. And deoxy forms, right? Is it likely that somebody could make a mistake on the right-on and put something oxygenated or deoxygenated when it was wrong?
Yeah.
Don't do that. Okay. Pay attention to what I'm asking you to write, whether it's RNA or DNA.
Okay.
Okay. It's just a memorization trick from there, and knowing how to connect the phosphodiester bond, which we will need to practice. Okay. So they're linked 5' to 3'. They are read 5' to 3'. Who else reads it 5' to, no, we're gonna read it the way we write it, 5' to 3'. Who else reads it 5' to 3'?
Enzymes.
The enzymes do. So that's probably a good direction to go. DNA's fairly stable. But RNA not so much. Some mRNA in cells degraded in a few hours. Some people who like to work with RNA are like, this stuff falls apart. It's like it's everywhere, RNAs. And they start accusing everything of RNAs just sitting around. Why is DNA more stable? Tell your neighbor, tell your group why you think DNA is more stable than RNA.
Because it's that dynamic.
Because of the deoxy.
Let's change the
Remember, it's on
hydrogen
the
bond.
second carbon, and that carbon is the one that's most often attacked. If you don't have a hydroxyl group, you don't have something that can form a lot of creative leaving groups. You don't have the ability to that second carbon as easily.
Isn't there like 2 of them? Do both of them have 2 strands? They're hanging out with each other? The DNA.
The hydroxyl group, if protonated, becomes deoxy and becomes a good base.
What? In what?
So by having it deoxy, so instead of a hydroxyl group, you just have a proton. It makes it less susceptible to attack from a chemical. That's how I think of it, anyway.
Okay. Let's take a look at some answers. We'll come back to that. Your goal is to be able to draw 5' to 3'. This is how hard it is for me to say that now that's expected. Boom, you're done. Okay, moving forward. Wait, 5' end right here had a hydroxyl, and I'm going to connect it with the 3'. And what am I going to sit in between it?
Phosphate.
A phosphate. Well, where did the that had a hydrogen. That had a hydrogen. Where did they go?
Water.
Water. Oh wait, this is another condensation reaction. It's like everything's like, yes, condensation reaction. It's called a phosphodiester bond. That's the name of it. Okay, and it's actually fairly stable in DNA. We can have some that's been around for a long time. I showed you Neanderthal. I got to meet the guy who sequenced Neanderthal DNA a couple times that he did. I also, woolly mammoth. Did you know we're trying to bring them back? Somebody's funded on that. They think it'll slow down global warming. Isn't that funny?
What?
Just to bring them back.
Yeah, we'll talk about it. But the fact that they go stir around with their tusks, the snow actually means that when it's wintertime, it actually gets colder to the permafrost and the tundra.
That'd be really cool.
The snow's insulated. Yeah, that's just so weird. So 5' to 3'. So if I listed ATG 5' to 3', that's how we're writing it. Okay? And someone will have a problem. If you haven't practiced it, it will not look pretty on the exam. And it will, you'll be like, I'm not sure if I can do this, right? Yeah.
So, I'm assuming that.
You'll have to put not an ATG.
Okay, so that was the.
Yes,
that's what I'm
yeah.
going
You're
with.
gonna have to put the actual
group,
Nucleotide.
right? Yeah, that makes sense.
Sorry.
So,
Yeah, that comes up, and I just am like, oh, well, why wouldn't I? That I just asked you to learn it. Yes, of course. You'll be writing it all. Thanks, Kevin, for asking. Because it's a real question, and it's a lot of work to have to memorize stuff. And then you wanna know.
And this way too, we'll write this way and, not this form, I guess this is the textbook, incorrect.
Yeah, you'll want it so that it, instead of the sin, it's in
Yes, sin,
the
yeah.
anti-position.
Gotcha, okay.
this is gonna be a crazy
Okay. Plants, sRNAs to prevent inbreeding, the hydrolysis of RNA, ribosome breaks it down with some RNAs, and dicer, all fun words for enzymes that chop up RNA, okay? Turns out that RNA is not stable, but why isn't it stable? And this is the answer, okay? And forevermore, when you're asked the question, you will answer this way instead of the way you probably just answered, okay? Most common answer, if I were to ask you a multiple choice question on this without any knowledge, is that you would say it's because it's double-stranded, because I get hydrogen bonding and it helps stabilize it. Does RNA actually bond to itself?
Yes.
Does it come in double-stranded?
Yes.
Does that make it any more stable?
No.
Nope. Okay, so that's not the right answer. What is it? It is this, when I have this in DNA, what is missing?
The hydroxyl group.
The hydroxyl, the very hydroxyl that if I have a nucleophilic attack here from a hydroxyl group or some other group that's ready, it'll reach out and grab that proton. Once it's grabbed that proton, you know I've got an electron-rich oxygen right there, right? And it's now actually physically next to the target, which is the phosphodiester bond. So it will move its electrons here, which will then mean too many electrons hanging around my phosphate, so it will actually kick it off, and that will cleave it.
So it cleaves itself, given enough time.
If it's in water.
Yeah.
Yes, it will.
Yeah.
So, and basic conditions will rapidly deteriorate it. So, Brother Rubish, Brother Lemon, and I consider this to be a fundamental principle in biochemistry. You're essentially establishing one of the class of molecules and why is it stable in its connection versus why is it not. And it's a chemistry question. And yes, it's organic in its nature of how we're describing that. We consider it so important that I have permission to flunk you if you don't answer this question right on the upcoming write-on on the exam. Did I get your attention? So you need to know this mechanism and get that answer right. Why is it unstable, RNA? And the answer is because it's got a hydroxyl on the 2', which allows it to undergo hydrolysis, breaking the phosphodiester bond. That's the words you say, right? But then you say, you draw out the mechanism as shown in the mechanism I just gave. Which would that be easy to draw if I had you draw RNA? As you can but I don't have to do that. I could just give it to you and then say, show me the mechanism. Or I could just assume you could draw it again. Okay? So that's what you're looking for. I don't need this part drawn, I just want the mechanism, because I want you to understand that, okay? If you want to take it to the product part, that's cool, but the mechanism is enough for you to understand it and for me to understand you know it. Okay? All right. Isn't that crazy?
That's kind of
Intimidating.
Did you flunk anyone?
Not yet.
Okay.
It's
like
but
there's
there's
still
some
time.
that came into second semester biochem and then didn't know it, and I heavily considered it right then and there. It's like,
Get out.
yeah.
Really? Wow. Man,
You're
embarrassing.
A pairs with T, C pairs G, C pairs with G. That means my adenine and my thymine getting together, my cytosine, my guanine getting together. Why is it that it does what it does? Well, let's examine this. This is how I make my hydrogen bonding between the two, so I get my double-stranded DNA. It could be RNA too, by the way. What am I looking at here? Hydrogen bonding. Okay. Why not here?
I think you're physically too far away.
It's too far. It's physically constrained. This is too far of a distance. Okay. Well, that sounds all right. And how many did I get here?
3.
All three.
I got 3. Oh. 3 because it's in what? Anti. Ooh. That's like important, isn't it? There will be instances where I will show you later on that we can have it being seen.
What?
And you'll see the effects of it. It's quite dramatic. And by the way, it probably exists in you and me. Frightening. We're living in sin.
What the?
Okay. All right. So that's hydrogen bonding. We will come back and talk about how that leads to what we call the major and minor groove. That'll be one of our focuses as we come back. Minor nucleotides do exist. So this is essentially akin to when we got to amino acids and we talked about that we could modify proteins, and we started talking about post-translational modification. We got to sugars, and what did we do with them? We said, hey, I can put all these decorations, these functional groups. Can I decorate my nucleosides?
Yeah.
Absolutely. You can put methyls on them. In fact, what happens when I put methyls on them?
Methylations.
These methylations have a big deal, right? This is epigenetic markers. Is epigenetics a big deal?
Yes.
This is the ability to potentially have the traits that even in some cases are heritable to the 3rd and 4th generation, just like in the Bible. You might actually be affecting what your children and your children's children and even the next generations, what their actual DNA says. Now, I'll use the one epigenetic story that most of you are probably familiar with. Somebody tell me about rats' mothers and epigenetics. Anybody know that story? Yeah, Logan?
With the mice, what they would do is they would take these mice and they would raise them under 2 different circumstances. One's where the mothers would groom them, take care of them, protect them, and others where the mothers were kind of negligent.
And it was rats in reality, but yes, keep going.
And what they found out was that the offspring that were taken care of were more likely to be able to, I believe that their DNA was more methylated and prevented certain cancers from being,
So more methylated, and it turns out that it's not about cancer, it's about behavior. Turns out that the pups raised without care, the methylations directly lead to an effect where they become more aggressive. They don't like to hang out with the rat pack. They live up in isolation alone. They're violent. They're mean. Those who were cared for and nurtured in their young we're able to do the opposite. They can live with the rat pack. They're more tame, so to speak. Don't reach too far. I'm sure your mother licked you well. But you have seen bunnies talked about at General Conference. You've seen, does it affect us whether or not you actually care for your children? Loving and, now I have no idea. It's much more complicated in humans, okay, to control those variables. But it turns out you can put implications on that. And we happen to be living in a society right now that is ever more what? Violent. Isolated. Because what are the parents doing? All day long.
Oh, wow.
How do we communicate with each other? If that's how we're communicating, is that a problem? We need to talk. Let's get contact. That's not to put you out of the comfort zone. Just care for your kids, you guys. All right. We'll come back and talk about this.
So